¿Qué tal? Buenos días. Soy Gabriela Parra del Instituto de Biología y estoy aquí para darles la bienvenida y agradecer su participación en esta serie de seminarios de Fronteras. El día de hoy le pedí al doctor Jorge Nieto del Jardín Botánico, también del Instituto de Biología, que fuera el moderador de este seminario, el cual va a ser impartido por la doctora Hake. Así que adiciono Jorge. Jorge, muchas gracias. Están en línea los dos. Hi, Sarah. Thank you very much.
Hi, Gabriela.
Muchas gracias, Gabriela, por darme la oportunidad de presentar a la doctora Sarah Hake. Well, it's a great pleasure to have you as one of our speakers in the seminar series Fronteras en Sistemática, Biodiversidad y Evolución, Frontiers in Systematics, Biodiversity and Evolution of the Institute of Biology at UNAM. It's actually a great way to start the International Women's Day, is today. Well, before you start your presentation, I'm going to take some minutes, Sarah, to give the audience a brief introduction about your scientific career. It is going to be in Spanish, so just be patient. It may take just 2-3 minutes. Buenos días, estimada audiencia de este seminario. Tenemos el placer hoy de contar con la doctora Sarah Hake, que es profesora de la Universidad de California en Berkeley, quien nos presentará el seminario El papel de los genes knox en la forma y diversidad de la hoja. Comentaba que hoy tenemos un excelente evento para iniciar el Día Internacional de la Mujer. La doctora Hake es líder internacional en el área de la biología del desarrollo vegetal, en particular en su análisis genético y molecular, como lo podrán apreciar durante su charla. A continuación les voy a dar una brevísima reseña de su amplia trayectoria científica. La doctora Hake trabajó por muchos años y hasta jubilación, que ocurrió hace poco tiempo, en el Plant Gene Expression Center del Departamento de Agricultura de los Estados Unidos en Albany, California. De este centro fungió como investigadora y como directora de 1998 al 2020. Y al mismo tiempo ha sido profesora adjunta del Departamento de Biología Vegetal Microbiana de la Universidad de California en Berkeley, puesto que sigue ocupando a la fecha. La doctora Hake estudió la carrera de biología en el Grinnell College en Iowa, el doctorado en Washington University en San Luis, Missouri, en el área de la biología vegetal, y posteriormente realizó un postdoctorado en la Universidad de California en Berkeley, antes de ocupar sus actuales posiciones académicas. La doctora Hake ha obtenido financiamiento para llevar a cabo sus investigaciones de varias instituciones. En su inicio, Pioneer Hybrid International, los Institutos Nacionales de Salud, NIH, y ha contado con muchísimo apoyo por muchos años de la Fundación Nacional para la Ciencia, el NSF, el Departamento de Agricultura de los Estados Unidos, a través de donativos competitivos del NRI y también del Departamento de Energía. La doctora Hake ha tenido muchos premios a lo largo de su carrera. Por ejemplo, en el 96 logró el premio Janet Ciron Pelton. En el 2008, el premio Stephen Hales de la American Society of Plant Biology. En 2009 fue electa como miembro de la National Academy of Sciences de los Estados Unidos. En 2009 también fue elegida como miembro de la AAAS, que es la asociación que publica la revista Science. En 2011 fue galardonada con el premio de científico senior del año del Servicio de Investigación en Agricultura del Departamento de Agricultura de los Estados Unidos. Y en el 2013 fue elegida miembro del Salón de la Fama de la Ciencia del Servicio de Investigación de Agricultura, o el ARS, del Departamento de Agricultura de los Estados Unidos. En docencia ha impartido cursos desde preparatoria en el Smithsonian. También durante muchos años ha impartido un curso a nivel de licenciatura en Berkeley de genética de plantas y biología molecular, y también un curso de posgrado sobre genética del desarrollo desde el 2007 al presente. Ha sido miembro de muchísimos comités dentro de la Fundación Nacional para la Ciencia, el Departamento de Agricultura, ha sido miembro directivo de las Maize Genome Database, ha sido secretaria de la International Society of Developmental Biology, es editora asociada de la revista PLOS Genetics, es miembro también de varios comités de la Academia Nacional de Ciencias de Estados Unidos, etc. Ha dirigido 20 tesis de doctorado y ha dirigido también a 30 investigadores postdoctorales que han estado en su laboratorio. Cuenta con una patente y ha publicado 143 artículos de investigación en revistas como Nature, Trends in Genetics, Genes and Development, Genetics, Plant Cell, Science, Development, PNAs, etc. Y los temas recurrentes de sus investigaciones son los siguientes, son todo biología del desarrollo vegetal, ha publicado sobre el genoma del maíz, sobre el estudio de caracterización de muchos mutantes en maíz, sobre el desarrollo de la lígula del maíz y mutantes que la afectan, el desarrollo de la hoja en los mutantes NOTED, que de hecho fue lo que la impulsó a seguir esta línea desde 1986 de genética del desarrollo y ha permitido lograr muchos hallazgos en este campo. En general es el análisis genético del desarrollo de la hoja, la iniciación y determinación de las hojas, también el desarrollo de la inflorescencia y su determinación, muchos estudios de genes homeóticos florales, el meristema apical del tallo es otro de sus temas de investigación, la gametogénesis femenina, la embriogénesis, genes que inducen hojas lobuladas en ciertas plantas como Arabidopsis. Ha también estudiado cómo se entrelazan el desarrollo vegetal y los mecanismos evolutivos. También ha estudiado la regulación de meristemos de la espiga en el maíz, el control de la filotaxia, la senescencia de las hojas, la forma e identidad de las células, la evidencia genética del origen del maíz, la inducción floral. Ha estudiado, por ejemplo, la iniciación de los primordios laterales y su relación con el rendimiento en los cultivos. Y últimamente ha estado haciendo trabajos muy interesantes sobre la síntesis del desarrollo de la hoja en las gramíneas. Y bueno, sin más, después de hacer este pequeño resumen de su extraordinaria carrera científica, le doy paso a la palabra a la doctora Hake para que nos imparta este interesante seminario. Thanks, Sarah, for keeping you waiting, and now I let you start your presentation.
Okay, thank you so much, Hake. And thank you, Gabriela, for helping me, and Ulysses also in giving this talk. So I'll get started now and tell you about a piece of work that I began, very early in my career and continue to do up until quite recently when I retired. And that's thinking about leaf diversity and leaf shape diversity. And I hear there's an amazing botanical garden at your institute or in your city. And of course, so most of you appreciate the beauty of plants as you walk through the botanical garden. And we have examples here of different leaf shapes where we have these water lilies that lay flat on the water surface to absorb light. This pea leaf here that is dissected or compound with stipules and leaflets and tendrils. And then a fennel leaf with its leaf base that is a delicious food that we love to eat, and these frilly fronds. And then an example of a...
Sarah, would you like to go full screen?
Okay.
We don't have you in full screen.
Right here. Does that do it? Is it now full screen?
Not yet.
Okay, for me, you and I are smaller on the side and it's bigger.
Yes.
I wonder. So in my screen, I can see it that way.
Okay. What if you click on the full screen button? Would that project the whole screen?
Okay, I can exit full screen. And then I can hit full screen again. This is in this platform.
Yeah.
The other thing I can try is to go to my presentation.
I think that's how you did it before. Yeah. Okay.
So how's that? Is that better?
Yes, thank you.
Okay. And in this case, I can't see you, but that's okay because I can hear you.
Okay, thank you.
So, just letting us admire the different shapes of leaves in this particular picture with the peas and its tendrils. And as Jorge said, I really love mutants. So this we know that these tendrils are also like leaflets because mutants will change the tendrils into leaflets, and other mutants will change the leaflets into tendrils. And fennel, with its leaf bases that wrap around the other leaves to make a food we like to eat, and carnivorous plants in which the leaf is modified to trap insects. So here's a brief outline of my talk. I'll first discuss where leaf comes from, and then explain proximal distal patterning in grass leaves, and then bring in knotted and how it's involved in proximal distal patterning. And one of the major key features of grass leaves in the proximal distal plane is the ligule, and we'll spend a lot of time talking about the ligule. And then I'll end by talking about knox genes in other species and how it has affected leaf diversity in those plants. So I think most of the audience is plant biologists, but just to remind us of the parts, we know that organs come from meristems, and the meristems are tucked inside the younger leaves in this dicot. These are axillary meristems in the axils of these leaves and these little axillary buds. And then in grasses such as maize, the meristem is very protected by outer leaves. And this is especially important because of the herbivory on grasses in the fields. And there are root meristems, and I know many of you probably work on roots, but I haven't, so I'm not going to talk about them. And we think about plants as being perpetually embryonic because of these meristems. They're always making organs. Here are 3 different views of meristems, and starting with maize on the right, we see this ball of cells as a meristem, and the P stands for plastocron. So there is a timing that plant leaves go through. So they go through different stages, just as a baby is a newborn, and then a toddler, and a child, and a teenager. The leaves all go through these stages. So every leaf starts off as a P1 and then becomes P2. So it's a way to describe leaf stages. And in this maize meristem, P1 is hidden back here because the leaves are coming off in opposite phyllotaxy. Tomato and ivy have a spiral phyllotaxy. So here's the P1 leaf, around here the P2 and P3. And you can see at this P3 stage, these trichomes are already developing. So depending upon the species, the differentiation occurs at different stages. The ivy meristem is useful to really think about the stem cells, which are right in the center, and the peripheral zone, or morphogenetic zone, as Don Kaplan called it. And so this morphogenetic zone is where the organs form. And we see this 0, and this is plastocron 0. And these are the cells that will be the next leaf. So the first question I want to ask is, well, how does a cell know that it is a stem cell in this red circle or a leaf initial cell? Clearly, the positions are different. One is more in the center, one is more on the edge, but how does a cell understand that position? And an answer to this question came from a dominant mutant called knotted. I will return to talk about this really amazing phenotype in a few minutes, but first I want to talk about the gene itself. So knotted is a homeobox gene, as I'll discuss later, and this is an in situ. And in this in situ, with the red line, the section was done right down the middle, revealing the meristem cut in the middle and the leaves that are initiating, leaf 1, plastocron 1, plastocron 2, plastocron 3. And here are our P0 cells, the cells that will make the next leaf. And they actually form a circle around the meristem. So we can learn 2 things immediately, that the cells that will become a leaf are missing knotted, whereas the cells that remain as stem cells are expressing knotted. We've also looked, this was work by Devin O'Connor, a graduate student, with the auxin transporter, and that's shown here on this picture. And the auxin transporter is a way to see the presence of auxin, at least auxin activity. And you see a strong signal here where knotted is low, auxin is high. And you can also see these opposite patterns by the high level of auxin in these veins that are developing, where knotted is missing in this major vein. So knotted is present where auxin is low, and knotted is absent where auxin is high. And so cells trying to determine where they are in the meristem, will they be a leaf or will they maintain as a stem cell, have these two cues. So I mentioned knotted is a homeodomain transcription factor, and just for someone in the audience who might not know what these types of proteins are, they bind to regulatory regions of genes and turn genes on or off. And I'll return at the end to talk a bit about knotted's function in this regard. So getting to the proximal distal patterning in leaves, we have, and this is true of all grasses, and if the grass doesn't have a ligule, it's probably a mutant. But we have tissue in the back here that's called the blade, the top part, and we have the sheath. And in this picture of Avena, we see how the sheath grips the younger leaves. So this leaf is older than this one, and it is really holding on tight with its sheath tissue. And here's the ligule shown in this picture here and here. And I think of the ligule as a sliding gasket. So what happens with the ligule is as the water runs down the leaf blade, it hits the auricle and spills off. And so it doesn't enter into the meristem region in the younger leaves. And the ligule also prevents insects from getting inside to the meristem. So this leaf has really formed a protective outer covering with the ligule. As I mentioned, all grasses have ligules, and there could be someone in the audience who knows of an exception, and please let me know. Now getting to maize specifically, I show these same pictures, but to remind you, the blade, the sheath, the auricle. And the auricle is really a tissue that allows the leaf to lean back. So maize leaves have a way to be upright and lean back, and it is the auricle that provides that flexibility. And we know that partly from a mutant, a couple mutants we have one that has a ligule but no auricle, and the leaves are upright. And leaf angle is really critical for crop yield. So as you can see in this dense field of corn somewhere in the United States, the plants are packed together very closely. And at the base of the plant, the leaves lean outward, but as you go up, the leaves are more and more upright, allowing them to be packed together and optimize photosynthesis. Lately, a number of papers have come out, looking at QTL for yield, and sometimes they are actually regulating the leaf angle. So the leaf angle has a direct implication in yield. But what I'm interested in is development and seeing the process of how the ligule forms. In this cartoon from Anne Sylvester, we see the different plasticron stages of these leaves. And in the beginning, at about a P2 leaf, it's just a little almost symmetrical shaped leaf primordia that becomes longer and longer. And the lines indicate patterns of division. The stipules are cell differentiation. So we can see there is a basipital trend of differentiation. And somewhere down here, when the leaf is still almost symmetrical, divisions begin to occur not right at the meristem, but about 50 microns away from the meristem. And the divisions occur in the longitudinal direction and the transverse direction. And you can see they're more intense here, and they're not accompanied by elongation. So these cells are dividing but not elongating, and therefore they're getting smaller and smaller. And then at a certain point, depending upon inbred, the little group of cells that have divided a lot pop out and make this little bump that will lead to a ligule. We can also follow the development of a ligule with the pin reporter, slightly different one than I showed before. This is a picture from graduate student Jian Moon when she was in the lab. And you can see, as you would expect, auxin is very strong. It's the yellow color in these lateral veins and the intermediate veins. Here's a slightly later stage. And at this later stage, we begin to see a little bit of the auxin signal on the epidermis in this line. And to me, this line is so fascinating because as with many things in plants, these cells making this line are only related by position, a certain distance from the top and a certain distance from the bottom. And here the ligule is probably becoming a bit more prevalent, but we see this line of auxin expression. So the ligule in this regard is also like a leaf initial. It sees a very specific expression of auxin. So now I want to move back to thinking about the dominant mutant, and I want to remind the audience that knotted is abundant in the meristem, absent in the leaves, and absent in the P0 region. And in the dominant mutants, this expression remains high here and off here, but it comes back in the leaves. And that's shown in this picture here, another in situ, where now the section is done above the meristem, up in the leaves, and we see little dots of knotted expression showing at the veins. And these dots of knotted expression lead to these striking veins. And I don't have the data for this in this picture, but these veins have cell signatures like auricle and sheath, tissue that is normally found more proximal. This is somewhat of a complex picture here, but I'll walk us through it slowly. And as I showed before, we've got blade, sheath is more proximal, blade is distal, and the auricle and ligule. And in knotted, here's a leaf that I've blown up here. Along the veins, because knotted is expressed in the veins, the cells become sheath-like, and they often have ligule fringes adjacent to them. So in many ways, the normally horizontal, the normally longitudinal proximal distal pattern is now recreated over and over in these veins, such that these sheath cells surrounded by blade cells can create ligules along their edges. So this was one of the first hints that knotted had a role in creating proximal distal patterning. Just to say two words, two slides, I give two slides about these dominant mutants. They're all due to changes in regulatory sequences. And maybe there are about 15 different alleles, often mutator elements, many of them in this big intron, some unknown thing that's as big as the intron itself, a DS element that allowed us to clone the gene, and an R-dotted element that we actually used, this allele we used a lot in our experiments. We also have loss of function alleles that I'll mention later, but in this case, the insertions of the transposons are in the coding regions. And it's curious to think about, well, how is it that the expression of knotted rearranges these tissues? And I think a hint comes from this slide from Katsutoshi Suda, a postdoc in my lab, that unfortunately we've never published this, but I find it interesting. And we're looking at chromatin marks in leaves. And normally, and B73 is our normal control, a maize inbred, and normally this marker of activation is very low across the knotted locus. So in a leaf, these leaves are not showing any epigenetic marks of transcription activation, but instead they are covered with marks of transcriptional repression, which is shown here as blue. And then if we look at the red, which is the R-dotted allele, we see a peak of activation right where that transposon is sitting, and we see that the repressive marks are low all across the locus. So our hypothesis is that the insertion of this transposon into an intron changes the chromatin marks and allows the gene to be expressed in the leaf. Thinking about proximal distal patterning, another allele gave us some very strong hints about knotted's role. And this was an allele given to us by Damon Lish, and therefore it's named DL. And it shows up late in the plant. So at about leaf 6 on the margins, we begin to see some little fringes. At leaf 12, we have these prongs, and these are like enations. And these sheath, they're sheathy, so the tissue is like sheath. Here's a blow up of one where it's paler green because sheath tends to be paler green. And with these veins now pointing into this enation. When we did in situs with this allele, we found a different pattern of expression. Knotted comes on in the margins and not in the veins. So the difference here is a different transposon, and I can't remember where it landed, but it changed the location of expression to the margins. And the last 3 leaves of this allele, the leaves are actually dissected. And so here's the third to the last leaf where it stops growing here. And there's a bit of an auricle in there, and the next leaf and the last. And this might be the only grass leaf mutant or allele that is actually dissected. And in this case, where we see these dissections, we see that knotted is expressed at the midvein in addition to the margins. So to summarize some of the in situs I've shown you, in wild type, knotted is not expressed at all in the leaves. In the Nufer allele with a dotted or dotted insertion, knotted comes on in the veins, and it gives those beautiful patterns along the leaf veins. In the knotted DL, we don't see an expression in younger leaves, and the expression here is inside the stem. You can go over here to see where I'm making these cuts. And so this is the stem and strong expression in those veins. And then as we move up into the leaf, we begin to see expression at margins. And we'll go to this one here where we see some stem, and here's a blow up of that, where we see the margin expression that will give venations and also very strong midrib expression. So I'll summarize the knotted data that I've told you about so far with this cartoon. Knotted is present in the meristem, and it's off in the cells that will make a leaf. And this leads to a normal leaf primordia where we still see knotted expression at the very bottom of it, but no knotted above it, leading to a normal leaf with regular blade, sheath, and the ligule auricle. In the dominant knotted mutants, one allele, the Nufer allele, knotted comes on in the veins. That creates sheath tissue and ligule tissue, auricle tissue along the veins. In another allele, knotted comes back on the margins and leads to these enations of prongs. And at the top of that particular allele, we see expression also in the midvein, and it prevents expansion and gives you another ligule, a new proximal distal patterning, but going down into the leaf. And very interesting in terms of this evolutionary series, a related animal protein also affects proximal distal patterning. And this protein is MEIS, mice, or mice, I'm not sure how they pronounce it, but this homeodomain protein related to knotted is expressed at the most proximal part of an appendage as it grows out, shown here. So it's responsible for this part of the limb. And so I find is that a coincidence or an evolutionary holdback that these two related proteins are affecting proximal distal patterning. Now, I've made a point, or tried to make a point, that knotted creates proximal distal patterning when it's misexpressed, and I now want to argue about its normal function in the leaf. And you could maybe imagine in this to-be leaf, the base of the leaf is touching knotted, and the tip of the leaf is not away from knotted. And we asked ourselves, what happens when knotted's missing? Is there a change in proximal distal patterning? And we had some alleles. This was the work of Eric Volbrecht, a student in the lab, and he found loss of function alleles. And depending upon inbred, they were shootless. Here's only a coleoptile in the knotted loss of function mutant. And here's a section made where this would be the scutellum, the coleoptile, and there's a flat region that would be normally a meristem and a P0 leaf. This phenotype is very similar to shoot meristemless, which is an orthologue in Arabidopsis. Here on the right is the STM mutant, only making cotyledons and no shoot, compared to the wild type that has the cotyledons. The meristem is in there. Yeah?
No, sorry, we can't see your pointer, so just so you know.
You can't. Okay, that's too bad. Okay. I will try to direct you with my words then. Sorry.
Okay, thank you.
Okay. So, but this phenotype was dependent upon inbred, and so some inbreds, the knotted mutants still grew and made inflorescences, although they were affected, making fewer branches, as shown in the left, or making very small ears, as shown on the right. So we wondered whether that was due to some redundancy. And knotted as are all the homeobox genes in plants, members of gene families. There are 8 in Arabidopsis, there are 12 in maize. And so we picked another maize knox-like gene to make a double mutant. And the one we picked is rough sheath 1, which was studied in the Freeling lab. And this one we knew was also expressed in the meristem, and that dominant mutants were due to the misexpression. So collaborating with a student in Mike Freeling's lab, we, Natalie Bolduc and my group, made the double mutants and found indeed, irregardless of inbred, if you lost both knotted and rough sheath, you had meristem arrest. And so we can compare panel G, which is B73, showing a meristem, and below it in panel I is the meristem and the leaves surrounding it. Whereas in panel H and the blow up in panel J, we see there's only the coleoptile and no sign of meristem or leaves in a flat panel, a flat area of growth. So getting back, my goal was to see how knotted affected proximal distal patterning. And unfortunately, we had no shoot rather than a shoot with a change in proximal distal patterning. So that made it hard to really confirm the role of knotted in proximal distal patterning. So then our lab took a focus on other mutants, trying to identify ones that lacked ligules. There are 2 classic mutants in maize that lack ligules, and one is ligulus 1, the other is ligulus 2. And ligulus 1 was actually found by the founders of maize genetics in 1910. So it's a very old mutant and more recently studied by Freeling and our group. But you can see in this mutant that the blade is still present and the sheath is present. The mutant is on the left, the wild type is on the right. But what you're missing is the ligule and the auricle. So this mutant can still make blade and sheath and have that differentiation. It just doesn't make the signposts of ligule development. Ligulus 2 is a little different. On the left is wild type and on the right is a mutant. And the ligulus 2 mutant does make an auricle and ligule at the very margins, leading to clear blade above it. But most of the leaf is just sheath-like, especially over the midrib. So this mutant fails to create boundaries in part of the leaf. And, you know, as typical geneticists, we made double mutants, and the double mutant is shown here compared to the normal SIB. So with a frontal view, you can see all auricle and ligule are missing, and the leaf is just somewhat wrapped up like sheath, quite the ways up. And on a side view on the right, we don't see any sign of a boundary, though I would say at the top of the leaf, it looks like blade, and the base of that leaf, it looks like sheath, but the blurring, the boundary is blurry. So the mutants were found, over 100 years ago, and the Freeling lab cloned them 30 years ago. And then a few of us have done in situs and immunolocalizations in the last decade. And this is an in situ from Robin Johnson, who I collaborated with. And we're going back to a picture of the meristem in the very center. And the red arrows mark the beginning sign of the ligulus 1 RNA, which starts at about P6, slightly before one sees an indentation, the actual outgrowth of the ligule. And the blue arrows indicate P7, where we now actually see a bit of a ligule. Mike Lewis in my lab developed an antibody and looked in more detail. And I've zoomed in on the region of P8 and P7 at the margins and the midrib. And what I want to point out here is how this antibody marks the nuclei of these cells that will become ligule. And you can see the pattern of protein localization is broad, and then it tightens up as the ligule forms. So it just becoming a denser and denser group of cells. And here's another immuno he took at a transverse section, showing that it is primarily epidermal in the very beginning of ligule development. Ligulus 2 has a similar pattern, and I'm pointing to the accumulation where there is a proper ligule that is visible to our eye, but we also see accumulation of this protein at the pre-ligule. So both ligulus 1 and ligulus 2 are showing up when these early cell divisions are occurring. Mike Lewis was interested in the expression of these genes across different species, so he used this Koji platform, which I hope most of you know about, to compare the genes and the orders of the genes in, starting at the top of this picture, ZMAs, then Brachy, Soteria, and sorghum. And you can see there's a lot of conservation at ligulus 1. There's also a lot of conservation at ligulus 2. And so he went ahead and tried his antibodies in these other grasses, and the maize antibody picked up the signal very, very strongly, showing accumulation in a well-developed ligule at the top of the middle panel, as well as at the base where the ligule is just beginning to form. So as I said earlier, all grasses have ligules, and they are probably all expressing a very conserved protein. And ligulus 2, Mike also looked at in Soteria and probably rice as well. And we can see accumulation of this protein at the ligule that's mature, as well as a broader region of expression in the pre-ligule. Now, we already knew about those two genes, so we wanted to find new genes. We always want new genes. And collaborating with Robin Johnson and Mike Scanlon, we did a laser capture experiment. She did most of the work. And in this experiment, she captured a 50 micron section of blade tissue, 100 micron section, or 50 microns of ligule, and 50 micron of sheath with a gap between the 3 different tissues, and then carried out RNA-seq of these different tissues. She also compared ligulus 1 and wild type, and then put all the data together in an analysis. And the genes that we focused on were these 31 genes. Now, these 31 genes are up in ligule compared to blade and sheath, and they were differentially expressed in the ligulus mutant compared to wild type. So they seemed like good candidates, expressed at the ligule, differentially regulated when the ligule was absent or not, to investigate in more detail. And we found a well, this is an unknown gene, so we just still call it unknown because it's unrelated to anything. And it is expressed at the ligule very strongly and not in other parts of the leaf. And you'll also see that it's expressed at the boundary of where the leaf initiates. Another gene was cuck-like, and that stands for cup-shaped cotyledon, a gene studied in Arabidopsis. It's expressed in the leaf at the region of the ligule and shown in J and K, but again, also in boundaries. And that made us go back and look at ligulus 1 and 2, where they also are expressed in boundaries, and sure enough, they are. Shown in I and J, we're looking at a transverse section through a tassel, and we see that the ligulus 1 antibody shows up at the junction between the main rachis and the tassel branch. And in J, we see that the protein shows up as a secondary branch is forming. G and H are longitudinal sections that also show this accumulation, but it's harder to make out what we're looking at. And indeed, we started looking at the tassel. I think for 110 years, people only looked at the leaf of ligulus 1 mutants, but we documented that there was a tassel defect as well with fewer branches, and they were also more upright in the ligulus 1 mutant. Annis Richardson, using a ligulus 2 antibody, the same one that Mike Lewis did, looked in the tassel, and indeed, it also, ligulus 2 also appears in the tassel, marking where organs form. And the fact that ligulus 2 mutants are often have a tassel phenotype, that was not new to everyone, but we have documented that, and we know this is inbred dependent. So in a separate experiment that I won't talk about, we've shown we're mapping QTL that regulate that branching. There was one more gene came out of the laser capture, And it was something we were already studying, and it's a bell-like homeodomain, short for BLH, bell-like homeodomain. And this one, this beautiful immuno done by Katsutoshi Suda, working with Hasmi and Abraham Juarez, who both were postdocs in the lab. Like knotted, this protein accumulates in the meristem and goes off when leaves are initiated. But unlike knotted, it in wild type comes back on in leaf bases, as we see in some of these panels. We see the RNA, and we also see a bit of protein on the outside of the leaves. Up in the upper right-hand corner is the control showing the antibody immunolocalization in a loss of function allele. And this protein was of interest to us because it interacts with knotted, and that interaction was shown very well by Hasmin in the lab in a paper they published together with Katsutoshi. And because this is an evolutionary talk, I wanted to bring up this old slide from Berglund, 1998, where he points out that knox and bell proteins in plants interact. And these are two, I've discussed these as well, but these interactions also occur in fungi and animals. And over in the animals is the MEIS, mice protein I mentioned earlier that functions in proximal distal patterning. So the homeobox genes are very old and very conserved, and they also have shared ancient interacting partners. So to summarize many of the things I've talked about and some things I haven't, I have this cartoon. We're looking at the expression of knotted and bell by itself, auxin, and boundary genes. And from the very beginning, I mentioned how knotted is expressed in the meristem, and its absence marks where leaves form, and the leaves are shown as that green semicircle in the meristem. And boundary genes are formed there. And these boundary genes come back on when we make ligules. In light blue in the left panel is the bell protein. So the bells, unlike knotted, are in the base of the leaf. And we also know that knotted protein moves into the base of the leaf because the protein can move through plasmodesmata. So our strong belief, maybe lacking all the data we need, is that these knox proteins set up the very proximal end of a leaf in terms of proximal distal patterning. And then the line is drawn, and we have boundary genes expressed at the ligule, and then leading to the very final form of a leaf with the distal blade, the proximal sheath, and the ligulin auricle. And what I've written down below is, I think I've said it, but just to reiterate, the meristem genes, I think, position the ligule. And then we know data that I haven't shown you, knotted binds ligulus 2, and we're pretty sure that ligulus 2 binds ligulus 1. So it isn't a simple linear line because other proteins come into play. But the boundary genes that define organogenesis are recruited again to make the ligule. Now, I want to spend my last minute or two, I know Jorge took up a fair amount of my time, but a few minutes to talk about what happens in other species. And an experiment we did years ago, Neelima Sinha and I did, we had hardly cloned knotted, and we transformed it into tobacco because that was what everybody transformed at that time. And we had an amazing result where on the left panel, instead of the big tobacco leaf, we had tiny, tiny shoots, and the shoots were covered with meristems. The leaves were viviparous or epiphyllous, and these plants never died. And so 20 years later, the plastic rotted, but the plants were still alive. Another experiment that Salamini's group, Muller et al., did was to clone the dominant mutant called hooded in barley. And in that mutant, little meristems form on the awns. And we did an experiment with Ross Williams Carrier and Peggy Lameau, where we transformed barley with knotted. And in the right panel, this is an image from our paper, where meristems, this is a floral meristem, formed on the awn, normally a leaf-like structure that would never have meristems. And another fun experiment we did many years ago that led us to think about cytokinin was to turn on knotted with a senescence-activated gene promoter. So SAG is senescence-activated gene. And a normal wild-type tobacco leaf the older leaves would start to die, as shown in panel A. But with knotted under control of this promoter, the leaves stayed green. And then in panel C and D, leaves were removed from the plant, and within 2 weeks, a normal tobacco leaf is starting to dry up, even on a wet paper towel. And by 4 weeks, it's very dead. But when knotted was turned on with the senescence-activated gene promoter, those leaves stayed green. Cytokinin was measured, and typically a young leaf in wild type, shown in black, has more cytokinin than old leaves. But in these transgenic plants, the old leaves had more cytokinin. So it was pretty clear that the presence of knotted in these senescing leaves led to an accumulation of cytokinin, which kept the leaves green. And another beautiful paper by Andrew Hudson's group looked at dominant mutants in Antirrhinum. And these two mutants, Hertzina and I don't know the names, Hertz and Ina, have these lobes on one of the petals, as shown in panel A. And when they cloned the genes, they found that transposons had turned the genes on, and these were in non-coding regions, just like in knotted. And it also transformed the shape of the leaf. So a simple transposon insertion changed the shape of the leaf, and it also gave petal lobes. And this makes me think about all that beautiful diversity in flower morphology with spurs and knot, and I wonder how often knox genes are involved. And tomato experiments were done by Eliezer Lifshitz, where they overexpressed knotted in tomato, leading to a super dissected leaflet form. And Neelima Sinha has carried on with a lot of this work as well. And I want to end with Miltos Siantis's work. Angela Hay was a student of mine, and they worked with cardamine, which is a dissected brassica, as shown in G. But when they knock down SDM, which is the ortholog of knotted, they lose the leaflets. And in a very recent paper by Miltos's group, they made mutants that really created leaflets in Arabidopsis. And I took these pictures from their paper, but if you focus on the black and white silhouettes, as in F, G, H, I, and J, F is wild-type Arabidopsis, J is wild-type cardamine, and in G, they've turned on SDM with a leaf promoter. In H, in G, they've put in a gene from cardamine called RCO, and in H, they have turned on SDM with a leaf promoter, and in D, they have added the two together and almost looking like the cardamine. So the leaflets aren't exactly perfect, but it is definitely a dissected leaf. 2 more slides of data just to remind you that it's a transcription factor. And we might wonder, well, how does knotted do all these amazing things in all these different species? And I think the answer is it regulates many, many genes and affects many hormone pathways. Experiments that Natalie Bolduc did with me a while ago, we dissected the regulatory region of an enzyme involved in gibberellin catabolism. When knotted is up, it turns on this gene and keeps GA low. And in blue, in the upper left panel, is the region where we had, with a lot of molecular biology, defined the region where knotted binds this gene. And then we carried out ChIP-seq, and she did this in 2012. And these sequencing reads overlapped her binding analysis very thoroughly. And in that paper in 2012, we also did RNA-seq. And you can see on the right how we've got modulated and bound. And modulated means the gene expressions are found to be differentially expressed. And we can see that auxin, brassinet steroids, gibberellins, ethylene are all affected. Interestingly, cytokinin didn't come up, but I think it could be because cytokinin is turned on and turned off. And when you look at a differential gene expression, you don't capture everything at once. I mentioned auxin in one of my very early slides, and with this picture, I want to show you the differential gene expression of auxin genes, looking at wild-type leaves ++, heterozygous leaves K+, and homozygous leaves. And I think this is a very nice illustration of how the dosage of knotted affects the function. And so you can see that knotted turns on genes with the red color, lighter in the heterozygote, stronger in the homozygote, and it turns off genes such as an OXIA gene in the middle of the panel, where it is a darker blue with a homozygote. So knotted does all the things it does in the leaf and in the meristem by regulating hormones. And it's pretty remarkable. So I think this is my last slide, and then I do some acknowledgements. I've mentioned these panels before, how knotted marks the meristem and goes off to make primordia, leading to normal leaves, and knotted comes back on in maize leaves, creating proximal distal patterns. And then in dicots, it also goes off to initiate a leaf, but in many dicot species, notably tomato and the brassicas that are dissected, it comes back on, creating leaflets. So this was a story created over, how many years was I working? 40 years, and many, many people involved, and I hope I've captured most of their names, but it was a lot of wonderful people to work with, students and postdocs, and constant funding from USDA, RS, which I'm very thankful for, and NSF. And so I can take some questions now, and I think I will put the screen.
Okay. Thank you very much, Sarah, for this very nice presentation, very elegant work. So there are several questions from the audience. Here we have the first one by Ulises Rosas. Amazing work. Thanks for this fabulous talk. In P, Uni, COCH and other genes are involved in leaf complexity. Have you found evidence of a role in leaf evolution in grasses?
That is cool. Uni is also called leafy, right? Uniflora, Ulises. I think it is leafy because I know that name, and it definitely has. There are really nice leaf. They're more architecture phenotypes. So I don't think there is a phenotype in maize, but it's a really great question. I'm going to write it down here and think about it. So a good question would be, did we find it in our laser capture, for example? I know that leafy is bound by knotted, but I don't know whether that regulation by knotted is part of its proximal distal patterning or something else. Nice question, thank you.
Okay, thank you for the answer. Another question by Ulises Rosas. Besides knox genes and UniCOCH, are there any other genetic mechanisms known in plants to control leaf complexity?
There's a gene called BOP, blade on petiole, in Arabidopsis, and there's a beautiful paper in rice where they knocked out 2 or 3 BOP genes and they end up without any sheath at all. So, and we know in maize that the BOP gene, in maize it's called true, but it is expressed specifically in the sheath. So there are genes, BOP genes, BOP, that regulate leaf complexity and that's also in Arabidopsis. So the BOP genes were first found in Arabidopsis, and they are expressed in the stipules, and they regulate knox genes. But if he doesn't know about BOP genes, he should definitely look there.
Great, thank you very much. Third question by Francisco Vergara Silva. Can you clarify your position on deep homology in plants and that goes to the next. If you think homologies between plants and animals can be established on the basis of the similarities you discussed, e.g. plant knox genes and mains in animals?
I've never heard the term deep homology, but maybe deep homology means going way back, plants and animals is, we'll assume that's the case. But what's very interesting, I think, is that these proteins partner in animals and plants, and these partnerships are very conserved. And in that slide, I mentioned an experiment in Chlamydomonas. And in Chlamydomonas, the knox and bell proteins have to interact to make zygotes. So it's really an amazing requirement for heterodimerization discovered very fundamentally in Chlamydomonas. And I think this is also true in plants, but plants have also, we have so much redundancy, our knox genes and our bell genes, that we don't get to narrow down on it. So I think there's possibilities there if you have an open mind.
Okay, thank you very much.
Thanks for that, Francisco.
Okay, the next question is by Alejandra Covarrubias, who you know.
I do.
Thank you so much for your great talk. Now questions. Which are the cues determining NOX1 protein expression and distribution? Are there any environmental cue involved?
I think this is really immune to the environment. I think this is so hardwired. I think the pattern of expression is going to be the same irregardless. But just as I say that, here's an experiment somebody did. A student in my lab looked at flax, and if you decapitate flax, a new shoot grows out of the stem, and those cells are actually expressing NOX genes prior to the decapitation. So in the stem, if you can imagine a stem of a plant, there are little pockets of NOX expressing genes ready to make meristems if they need to. So it is possible that environmental cues such as decapitation or wounding lead to some new NOX expression. But I don't think they are affected by things like cold or heat or those sorts of environmental cues. I think she must have an idea, which is why she's asking that. I don't know.
Okay.
Nice question, though.
Yeah, this reminds me what you said about a paper that just came out by Rodit Sowers about local adaptation in land races of Mexico, the Palomero Toluqueño. And he found that the spike, the male inflorescence,
the tassel, yeah,
it's very neutral to environment changes. Like he did the common garden experiments of different segregations of E73 to Palomero Toluqueño, and basically the spike is morphology and everything is the same in the highlands or in tropical areas.
I believe.
Whereas other things change very much. Like of course, yield components like you know the ear size, ear number of kernels, stuff like that. So,
that's cool. Yeah, I'll have to look at the paper. Rory's paper?
Yes. So you think that then there is like these compensation mechanisms, like temperature compensators and things like that, like in circadian rhythms that control or that maintain the shape of some of these developmental pathways independent of the environmental conditions.
Yeah. You know, so many genes are under circadian clock control, but I wonder if the NOX genes are not, you know, so maybe they are outside of that sort of circadian control or light dependent control. And just like the embryo that is developing, you know, in an animal, the meristem is developing really hidden by, you know, the environment and maybe protected in multiple ways. maybe transcriptionally, it's just the genes are not able to fill these environmental changes. But I think if you were to look at somebody's heat shock experiments or cold or drought or all those kinds of experiments, you wouldn't find knox genes on their list of differentially expressed genes. But it is a good question. How are they protected from change?
Yeah. Well, I have a question, Sarah. I work with this stuff of seedling emergence in maize, and this relates to the coleoptile. So the question is, can you comment on the relationship between leaves in grasses and the coleoptile? Basically, the question is it a leaf or is it a cotyledon, as some people have said? What represents the coleoptile? Is the missing cotyledon of the monocots? In other words, how homeotic genes affect coleoptile, and if they do they do it similarly to a leaf?
Well, Kaplan would say that it's the leaf base of the scutellum, so that, there's the scutellum and the coleoptile, and this is really just part of that scutellum. But knox genes don't affect it. So you remember my slide that
right. That's what I.
It has a coleoptile.
So basically, you would say that the coleoptile is not a leaf.
I would say it's not a leaf.
So it's more like a cotyledon. So some monocots are dicots.
Well, it's not
with derived. A weird cotyledon.
Yeah, it's not derived from the meristem. So, the meristem. So we have some pictures Dave Jackson did of a very small embryo in maize, and knotted begins. That embryo is there, and then knotted begins to be accumulating on the side of it, of the embryo, after it's formed. And so this scutellum exists without knox genes, and it must have been formed by the mother somehow, So I would say a coleoptile is not like a leaf.
Right. Okay. Thank you very much. Oh, there is another question by Joseph Dubrovsky. Hi, Sarah. Beautiful work. What are the common mechanisms of action between knotted genes in Hake in animals?
Hake and obviously in animals, limbs are very, very complex, and not the Hake genes are doing that. But in our very, very simplistic leaves, where we just have these primordias leaving the meristem, I would just say the common mechanism is setting up proximal distal. So proximal meaning adjacent to the meristem, and distal away from the meristem. So that is maybe the only. And then the common mechanism, they have the very same binding motifs, and the binding domains are the same. Highly conserved amino acids. I can't remember them now, but I used to memorize them. So the binding of knotted to DNA is highly conserved with animal related proteins. So the heterodimerization is conserved, the binding is conserved, and there's a little common mechanism in terms of their output. A little bit.
Okay.
So I guess that's our last question, Sarah. That was our last question.
Okay,
so
great.
Thank you so much. Thank you.
Yeah, and so I'm going to go out, and then I'm going to come back in with that other link.
Yes.
Okay.
Y muchas gracias a la audiencia por su participación, por su presencia, y no se olviden nuestro próximo seminario, que es el 22 de este mismo mes. Muchas gracias.
Okay, bye-bye, Sarah.
Thank you, Sarah.
Thank you very much.